Hello, future doctors and scientists. Welcome to an exciting journey into the microscopic world. Today, we're diving into one of biology's most fundamental and fascinating processes, a cornerstone of developmental biology. We're going to explore a process that happens inside every one of us. It's called gastrulation. This is where the magic truly begins. Imagine a construction crew, a highly skilled team ready to build something amazing. They're arriving at a building site, not just any site, but one where the very foundations of life are about to be laid. Similarly, gastrulation is when a simple clump of cells, seemingly unorganized, gets organized in a spectacular fashion. It's like they're meticulously laying down the blueprint for the entire body, deciding where everything will eventually go. Before gastrulation, the embryo is just a hollow ball of cells, a seemingly simple structure. This hollow ball is called a blastula. It's the starting point of an incredible transformation. Afterward, it transforms into a multi-layered structure, a complex and organized entity. This new structure boasts a basic gut and defined body axis, setting the stage for further development. This process is absolutely essential for every animal on the planet. It's vital for any animal with more than one tissue type, and yes, that includes us humans. The precision of these cell movements is astonishing, a testament to the intricate choreography of life. And because it's so precise, even small errors can lead to major developmental issues. By studying gastrulation, we can unlock secrets about birth defects, gaining insights into how they arise and how to potentially prevent them. We can also learn about tissue regeneration, understanding how bodies can repair and rebuild themselves. And incredibly, even cancer, as the process has gone awry in cancer development, often echo those seen in early embryogenesis. Over the course of this chapter, we'll explore the three germ layers, the primary tissue layers of the body. We'll see how these layers form during gastrulation and what organs and tissues they eventually give rise to. We'll also delve into the dance moves cells use, the intricate patterns of migration and signaling that guide their development. And finally, we'll examine how different animals accomplish this feat, revealing the diverse strategies that evolution has crafted. My goal is to make this complex topic simple, visual, and exciting, so that anyone can understand and appreciate the beauty of developmental biology. By the end, you'll not only be ready for your exams with a solid understanding of gastrulation, but you'll also appreciate the elegant science that shapes all of us, the intricate processes that transform a single cell into a complex organism. So let's get started, because science is undeniably cool. So what exactly is gastrulation? It's a fundamental process in the development of all animals, a critical transition that shapes the future organism. It's the phase where a relatively simple, single-layered blastula dramatically reorganizes itself into a more complex, multi-layered structure called the gastrula. This transformation is key to establishing the body plan. Picture the blastula as a hollow soccer ball, a sphere of cells with a fluid-filled cavity inside. Gastrulation transforms this simple ball into a complex structure with distinct internal layers. These layers are the foundation for all the different tissues and organs of the developing animal. Cells migrate in a highly coordinated fashion from the surface of the blastula to the interior, changing positions and fates. This inward movement and reorganization is what leads to the creation of the foundational layers of the body, also known as the germ layers. The term gastrulation itself comes from the Greek word gaster, which means stomach. This is because the primitive gut, or archenteron, forms during this crucial process. Gastrulation also establishes the body's major axes, the head-to-tail, anterior-posterior axis, and the back-to-belly, dorsal-ventral axis. Furthermore, gastrulation defines an inside and outside for the embryo, setting up the basic body plan. After gastrulation, cells are essentially committed to their future identities. Their developmental paths are largely determined. This commitment is significant. It's often considered the point of no return in embryonic development. The entire body blueprint, the fundamental organization of the organism, is essentially laid out during this relatively short but incredibly dynamic period. In summary, gastrulation is the process that turns a simple ball of cells, the blastula, into a more complex embryo, the gastrula, complete with a basic gut and defined body axes. This transformation is driven by highly coordinated and precisely orchestrated cell movements. These movements ultimately set up the three primary germ layers, the ectoderm, Mesoderm, and Endoderm. These germ layers are the raw materials, the foundational building blocks for every tissue and organ in the developing organism. Without gastrulation, this fundamental process, complex animal life as we know it simply wouldn't exist. It's a cornerstone of multicellular organism development. Let's meet the three primary germ layers: Ectoderm, Mesoderm, and Endoderm, the primary colors of development. The ectoderm is the outermost layer. It forms the skin, nervous system, hair, nails, and the lenses of your eyes. Next, the mesoderm, the middle layer, creates muscles, bones, the circulatory system, kidneys, and much of the reproductive system. Think of the mesoderm as the body's stuffing, filling the space between skin and gut, providing structure and movement. Finally, the endoderm, the innermost layer, forms the lining of the digestive tract, liver, pancreas, lungs, and respiratory tract. The endoderm manages the body's internal environment, handling nutrient absorption and gas exchange. These three layers interact to build every tissue and organ in the body. Understanding what each layer becomes is key for exams and for appreciating how the body is built. From these simple layers, the complexity of life unfolds. During gastrulation, a remarkable transformation occurs. Cells, once part of a simple structure, begin a complex journey. But how exactly do these cells navigate and move to their designated locations? It's not chaos, far from it. Cells use coordinated dance moves, a carefully orchestrated series of movements that ensure each cell arrives at its correct destination. This choreography is essential for proper development. There are 5 main types of these cellular dance moves: invagination, ingression, involution, epiboly, and delamination. Each one plays a crucial role in shaping the developing embryo. Invagination. Imagine a sheet of cells folding inward, like poking a finger into a soft ball, a process beautifully seen in sea urchins. This creates an inner layer within the developing embryo. Ingression. Here, individual cells break away from the main sheet and migrate inside the embryo, like people leaving a crowd and venturing off on their own, also prominently seen in sea urchins. Involution. In this movement, a sheet of cells rolls inward, creating a new layer as it turns. It's like a conveyor belt smoothly transporting cells, a key process in frog gastrulation, ensuring proper tissue formation. Epiboly. This involves a sheet of cells spreading out and thinning to enclose the deeper layers of the embryo, much like pulling a beanie over your head, a process commonly observed during ectoderm formation, the outermost layer. Delamination. Here, a single sheet of cells splits into 2 distinct layers, effectively creating new tissue layers, a process frequently seen in the development of birds and mammals. These movements are the fundamental developmental toolkit, the essential set of actions for building the complex bodies of animals. Each animal employs a unique combination of these movements, but the underlying goal remains constant: to meticulously organize cells into the 3 primary germ layers: the ectoderm, mesoderm, and endoderm. Understanding these intricate cellular movements is crucial, as it helps explain the remarkable diversity we see in animal development across the biological world. Let's see how different animals use these moves. Sea urchins with little yolk use ingression and invagination to form their germ layers. Frogs, with more yolk, use involution and epiboly. Cells roll over the blastopore and spread to cover the embryo. Chickens, with massive yolk, form a flat blastoderm and use a primitive streak for cells to ingress and involute. Zebrafish combine involution and epiboly around the blastoderm margin to engulf the yolk. Humans, despite little yolk, use a primitive streak like birds and reptiles, reflecting our evolutionary history. Evolution adapts the same basic processes to suit different eggs and life histories. The choreography may differ, but the outcome, a multi-layered embryo, is universal. Why does gastrulation matter for you? It's a cornerstone of developmental biology and a favorite exam topic. Knowing which tissues arise from which germ layer is essential for exams like NEAT or NET. Beyond tests, errors in gastrulation can cause birth defects, like serenomelia or situs inversus. Studying gastrulation helps us understand these conditions and the genetic signals behind them. The process also offers insights into cancer, where cell migration and organization go awry, and regenerative medicine, where we try to guide stem cells to form tissues. Gastrulation is the instruction manual for building the body. Mastering it gives you a foundation for anatomy, physiology, and medicine. It's not just important, it's essential for understanding life and health. We've journeyed from the blastula to the gastrula, met the 3 germ layers, and watched the cellular dance that builds life. Visualize these processes, draw diagrams, use colors, and watch animations to make the concept stick. Science is a process of discovery. There's always more to learn about how cells organize and build us. Stay curious, ask questions, and build your knowledge layer by layer, just like an embryo. Embryology can be challenging, but it's incredibly rewarding and reveals the elegance of life. You're learning the story of how we're made, written in the language of cells and genes. Keep exploring, keep visualizing, and keep being amazed by the science of life. The adventure is just beginning. Thanks for joining me today. Now go out there and learn something wonderful.